This is Randy Shell, and I'm making a podcast on the topic biochemistry and metabolism in anesthesia. This podcast is part of the University of Kentucky Department of Anesthesiology didactic curriculum. Let's first look at the ABA content outline and some recommended readings that I've provided. In the content outline, biochemistry of normal body metabolism, focusing on carbohydrates and proteins, and organ metabolism of the brain, heart, liver, and muscle, is where the questions on the American Board of Anesthesia exams come from. The recommended readings come from Miller's Anesthesia, 8th edition, 2015, chapter 106, and Stolting's Pharmacology and Physiology, 5th edition, 2015, chapter 33. I have developed some select topics around biochemistry and metabolism using the American Board of Anesthesia in-training exam keywords on biochemistry and metabolism from 2008 through 2017. Those can be divided into the major topics of carbohydrates, stress response, respiratory quotient, and some miscellaneous topics such as cyanide and its effect on metabolism, brain and its use of ketones, septic shock, and some of the metabolic effects of it. One of the gaps in knowledge, is where most people who took the exam for their CA3 year missed the question, was the topic of lipogenesis being associated with a higher respiratory quotient than gluconeogenesis. And we'll touch on respiratory quotients and metabolism. So let's move into our outline of topics. We'll start with carbohydrate metabolism, an overview, surgical stress response and what happens during surgical stress, and then go into select topics, briefly touching on respiratory quotient, fasting metabolism, metabolic syndrome, cyanide toxicity, sepsis, and metabolism and biochemistry of sepsis, and then finish up with some self-assessment questions. Let's go into carbohydrates first. Glucose is the predominant molecule to produce energy for our body. ATP is made from glucose, and glucose is the source of energy for our brain. In fact, the brain needs glucose. Yes, it can use a little bit of ketones, but the major sole energy source for the brain is glucose. Sugar is transported through cell membranes. It's protein carrier mediated diffusion and provides an immediate energy source, or it can, in states of high glucose and not necessary energy demands, that glucose can be formed into glycogen. Glucose movement, transport through cell membranes inside, is enhanced by insulin, and there is a resistance to this movement of glucose into cells in the case of non-insulin dependent diabetics, and also during sepsis, and the result is often hyperglycemia. Hyperglycemia has some bad side effects, including immunosuppression, stimulation of the sympathetic nervous system, increased infections, decreased wound healing, and when you have a lot of sugar presented to the kidney, an osmotic diuresis can occur. In the graphic on the far right shows when food intake occurs and blood glucose goes up, there's several things that can happen. In the case of high blood sugar, adipose tissue, fat can be formed. In the case of when the blood sugar is down, fat can be metabolized and ketones produced. Blood sugar is needed in the brain. Blood sugar can go to the liver and form glycogen, or glycogen from the liver can be broken down into glucose when it's needed. Pancreas produces insulin, which drives glucose into cells, and the muscle can use glucose and has glycogen stores also. So the liver regulates, stores, and produces glucose, and glycogen itself, which is multiple glucose molecules linked together, is mainly stored in the liver but also in the skeletal muscle. The formation of glucose from amino acids and the glycerol portion of fat is called gluconeogenesis, or formation of new sugar. And in the states of hypoglycemia, when cortisol levels go up, cortisol can mobilize proteins, and when blood sugars are down, new sugar can be formed from other substances. If you look at glucose and how it goes through the metabolic cycle to form energy, we can go all the way back to biochemistry in college or even high school, where glucose goes to pyruvate, and under conditions of oxygen, adequate oxygen, aerobic metabolism occurs via the Krebs cycle. And through oxidative phosphorylation in the mitochondria, we have the production of ATP and NADPH, energy for our cells. And under anaerobic conditions, glucose goes to a lactate, and metabolic acidosis can occur. So under normal aerobic conditions, when we use glucose, it goes into the Krebs cycle, it goes into oxidative phosphorylation, and we get energy production. When there's not enough oxygen around, we can get anaerobic metabolism to lactate. When we get oxygen back again, under aerobic conditions, a well-functioning liver can take that lactate and convert it back into glucose. Oxidative phosphorylation, that energy production, our energy-producing factories in our body are in the mitochondria, and cyanide is unique in that it can poison that oxidative phosphorylation process, or the energy production, and tissues no longer can use oxygen, and tissue hypoxia occurs, and acidosis, and cyanide poisoned patients can die. Brain mainly uses glucose, and it's basically the sole energy source, although it can use some ketones. When you have a brain that is hypoglycemic, obviously that's a huge risk for damaging a brain, but hyperglycemia is also not good for an ischemic brain because it worsens acidosis. And so when we have brain ischemia and hyperglycemia concomitantly, we often try to keep the blood sugars down into more reasonable levels because it can worsen acidosis and worsen ischemic brain injury. There are some hormones that affect carbohydrate metabolism. Insulin goes up during an anabolic state, that is when you're building things and not breaking down things. And insulin tends to go down during surgery, during stress. There's insulin resistance, and so during stress response and during perioperative surgery, oftentimes we see hyperglycemia. Glucagon is a catabolic hormone, comes from the pancreas, increases cyclic AMP inside cells, and stimulates the liver to break down glycogen to glucose and to form new sugars. And so glucagon raises blood sugar and can be used actually to treat patients who are hypoglycemic. One of the other unique uses of glucagon is in patients with beta blocker overdose. Glucagon increases intracellular cyclic AMP, and it does this not requiring beta receptors, for example. It's a non-receptor mediated effect. Beta blockers, obviously, by attaching to the beta receptor outside the cell, can reduce intracellular cyclic AMP, and glucagon can reverse some of these effects. Glucocorticoids also have effect on carbohydrates. Glucocorticoids come from the adrenal cortex and go up with stress. These glucocorticoids cause the formation of new glucose and sensitize the adipose tissue to lipolysis and increase proteolysis. And so basically, when your stress hormone glucocorticoids go up from your adrenal, your sugar's going to go up, your fat's going to break down, your protein's going to break down, because your body needs sugar to meet that massive stress response that is occurring. Etomidate is one of our hypnotic induction agents that is unique in the fact that it can block adrenal steroidogenesis, and that's shown in the graphic on the far right, where cholesterol is going on the left side down to form cortisol, and on the right side of the graphic, cholesterol is going down to form aldosterone. So the glucocorticoid pathway on the left and the mineralocorticoid pathway on the right. Etomidate affects several enzymes, one of the important ones being 11-beta hydroxylase. If it blocks this enzyme, which it does, there can be a reduction in glucocorticoids, in mineralocorticoids. And we usually think of mainly the glucocorticoids and often forget that mineralocorticoids often can be affected also. Other hormones, human growth hormone goes up with stress, and that promotes, again, new sugar and promotes lipolysis or breakdown of fats, and epinephrine, because through its beta-2 effect, raises blood sugars, and several reasons why it does that. It promotes the breakdown of glycogen, the formation of new sugar in the liver. It results in glucagon release and decreases insulin secretion, and all those things together result in a hyperglycemia when epinephrine levels go up, and that's classic in the surgical stress response. In the surgical perioperative period is to have patients with hyperglycemia. Insulin, some of the actions are shown at the top right. Let's look at those first. Insulin causes glucose uptake into muscle, into adipose tissue. So when our blood sugar goes up, our insulin tends to go up and drives the glucose into muscle and into adipose tissue. Glycolysis is increased, glycogen synthesis is increased, so you're taking singular glucose molecules and converting them into glycogen. Protein synthesis goes up, and potassium is driven into the cell. Stress actually decreases insulin production, and during stress, such as in the perioperative surgical period, again, hyperglycemia frequently occurs. Starvation and uncontrolled diabetes mellitus, some of the things that can happen there: lipolysis and fatty acid metabolism can occur. Epinephrine and norepinephrine mobilize fatty acids. Energy for our body can occur from metabolism of lipids during these starvation states and during uncontrolled diabetes. And we can get these ketones, acetoacetic acid, beta-hydroxybutyric acid, and acetone, and resulting in a ketotic state or ketosis. So starvation and uncontrolled diabetes can result in ketosis. Let's look at the stress response now, starting with a wound, such as a surgical incision. With a wound, local cytokines are produced, nerve signals are transmitted to the spinal cord, sympathetic nervous system input to the brain and output to the body, and the brain produces ACTH, or ACTH from the pituitary gland, and that goes down to the adrenal and says produce cortisol and epinephrine. So ACTH is a stress hormone. Cortisol and epinephrine result stress hormones. Glucagon goes up also from the pancreas, and as you can see, if you have lots of cortisol and lots of epinephrine and lots of glucagon, you expect during that stress response to have a hyperglycemic state, and that is what frequently occurs. The stress response shown up at the top causes the hypothalamus and the pituitary to act. Nerve signals go down to our adrenal medulla, where epinephrine and norepinephrine are released, and that results in the short-term stress response, the effects of epinephrine and norepinephrine. Glycogen is broken down to glucose, raising our blood sugar level. Our blood pressure goes up. We breathe faster. Metabolic rate goes up. And blood flow is diverted to areas of our body for response to the stress. That's the short-term stress response. mainly epinephrine and norepinephrine mediated. Also, however, the hypothalamus and pituitary under stress result in the release of ACTH, which at the adrenal cortex causes secretion of mineralocorticoids and glucocorticoids, which are part of the long-term stress response. The long-term stress response mediated by mineralocorticoids results in retention of sodium and water, and our volume in our body goes up, and our blood pressure often will go up also. Glucocorticoids result in protein and fat being broken down and converted to sugar, and so blood sugar goes up, and cortisol also suppresses the immune system. So the short-term response, think of it being mediated by epinephrine and norepinephrine. The long-term response of our stress being mediated by mineralocorticoids and glucocorticoids. Stress response increases many hormones. As mentioned, cortisol goes up, catecholamines goes up, glucagon goes up, ADH goes up, and ADH, or vasopressin, results in vasoconstriction as well as water retention. Growth hormone goes up with glucose intolerance, and the overall result of the stress response is one of catabolism, that is breaking down things. A negative nitrogen balance occurs, and the substrate for increased hepatic gluconeogenesis can be protein. Protein catabolism, and as you break down protein and get into this hypermetabolic state, you can get hyperglycemic. You also can get increased lipolysis or fat breakdown. So the metabolic response to stress is catabolism of protein and breaking down of fat and blood sugar going up. Ketones. Ketones are produced in diabetics, for example, patients on ketotic diets, which are low in carbohydrates, and patients who are fasted for extended periods of time. At the top right shows 3 of the ketones: acetone, acetoacetic acid, and beta-hydroxybutyric acid. These ketones are produced by the liver from fatty acids in times when there's not a lot of food around or there's not a lot of carbohydrate around. So the graphic at the bottom right shows the liver taking fatty acids, for example, converting them to acetyl-CoA and to acetoacetic acetate and beta-hydroxybutyrate. The brain and the heart can use ketones for some of its energy, but the brain also has an obligatory requirement for some glucose. So the main energy source of the brain is glucose. However, it can use some ketones. Interestingly, ketones cannot be used by the liver for energy. However, those ketones can go to other places in the body and be used for energy. Let's look at a couple topics. The first one being respiratory quotient. Respiratory quotient, by definition, is the volume of CO2 produced over the volume of oxygen consumed, for example, when you metabolize a carbohydrate. And that's the example at the top right in yellow of glucose with 6 carbons. Under oxidation with 6 oxygen around, it forms carbon dioxide and water. And notice that if we take the volume of CO2 produced, which is 6 molecules, over the oxygen used, which is 6 molecules, that ratio of 6/6 is 1, and the respiratory quotient of carbohydrates is 1. Palmitic acid is an example of fat metabolism, and in this case we have fat, palmitic acid, joining with oxygen, 23 molecules, forming 16 molecules of carbon dioxide and some water. And if we take the ratio of the CO2, which is 16, and divide it by the amount of oxygen that was used, 23, that ratio is 0.7. And therefore, the respiratory quotient of fats tends to be about 0.7. The normal total body respiratory quotient, if we add up all the nutrients that we tend to use, the normal is about 200 over 250, or approximately 0.8 respiratory quotient. So if we look at the table here of energy expenditure and caloric requirements, you can see that the respiratory quotient for carbohydrates is approximately 1. For fat, it's 0.7. For protein, it's 0.8. And we said the average for all nutrients is approximately 0.8. If lipogenesis is occurring, that is, you're actually having so much caloric intake that you're forming fat, the respiratory quotient can be greater than 1. And one of the things in the ICU that can be done occasionally is measuring respiratory quotient and seeing if you're overfeeding a patient, for example. And so net lipogenesis can occur when the respiratory quotient is greater than 1. The next topic is fasting. And with fasting, you need energy, obviously, and if you're not taking in food, where can it come from? Well, it can come from glycogen stores that are broken down. That's glycogenolysis, and that is supported by increased glucagon and decreased insulin. In fasting, cortisol levels go up and catecholamines go up. It's a form of stress state. Catecholamines stimulate beta-2 receptor, and that beta-2 adrenergic stimulation accelerates lipolysis or breakdown of fat. So while you're fasting, you can get sugar from breakdown of glycogen. You can get a breakdown of fat. And with that increased lipolysis and formation of triglycerides and free fatty acids, the triglycerides going to free fatty acids and glycerol, some of those free fatty acids can be converted in the liver to ketones for energy. So lipolysis, glycogenolysis, and if there's prolonged starvation, the body needs energy from somewhere, and if there's not fat and glucose around, it can break down muscle. So skeletal muscle and smooth muscle can be broken down to amino acids, and that can be a substrate for gluconeogenesis. In a catabolic state, muscle breakdown state, negative nitrogen balance state can occur. Metabolic syndrome is very common in the world's populations. Estimated that about a quarter of the population have this syndrome that is made up of obesity, dyslipidemia, hypertension, and hyperglycemia. Just think of the patients that maybe you have taken care of in the last week or so. How many of them have these components of overweight, lipid problems, hypertension, and high blood sugar? And this is associated with cardiovascular risk, type 2 diabetes, increased perioperative risk itself. And the treatment is diet and lifestyle changes. There are some criteria that are used to make this diagnosis of metabolic syndrome. Any three of the following characteristics, according to Stolting's Physiology and Pharmacology 2015 textbook. And that is the waist circumference, the blood glucose concentration, the increased systemic blood pressure, the serum triglyceride concentration, and HDLs. Cyanide toxicity is the next topic, and the reason why this is being touched on is because of the effects of cyanide on our metabolic systems. Cyanide can be formed when there's incomplete combustion of some energy source, such as in the car, burning gasoline, a heater, smoke inhalation where fires are burning. Cyanide can be produced. And usually we think of incomplete combustion of carbon monoxide, that is the major thing produced, but also in fires, cyanide can be released. So someone who is pulled from a fire, for example, may not only have carbon monoxide poisoning, but may also have cyanide toxicity. What does cyanide do? Well, cyanide poisons the cytochrome oxidase system, and oxidative phosphorylation is paralyzed. So up at the top right is showing the cytochrome oxidative system or oxidative phosphorylation, and we have cytochrome A3 being poisoned by this, and therefore oxygen cannot be used to produce energy, and you have tissue hypoxia. that happens. Cyanide in the perioperative period can occur when nitroprusside is being infused. ICU patient, for example, with very bad hypertension, trying to control blood pressure with nitroprusside in high doses. You can suspect it when nitroprusside is infusing and the dosage is having to be increased to get the same blood pressure effect. They're becoming tolerant to the effects of nitroprusside, and you draw a blood gas and there's a metabolic acidosis present, lactate building up, but mixed venous may be increased. Why? Because if your tissues aren't using the oxygen just goes to the tissues, comes right back to the lung and in the main pulmonary artery. If you measure mixed venous, instead of being maybe in the 70% range, it could be way up into the 90s. You're just not using the oxygen. So cyanide blocks oxidative phosphorylation. The tissues cannot use oxygen. Acidosis develops, and the patient can die very quickly from cyanide toxicity. The last topic is sepsis, and what are some of the metabolic and biochemical changes that occur with sepsis. Septic shock can cause catecholamine-related increase in the rate of glycolysis. Your catecholamine levels go up with the stress of sepsis. With those catecholamines stimulating cellular metabolism, cellular ATP demands may increase beyond the capacity of cellular oxygen supply. And if you have ATP demands more than what cellular oxygen supply is present, then you may have anaerobic glycolysis and the formation of lactate. Septic shock also can result in mitochondrial failure, and some of the things that are released during sepsis, like cytokines and bacterial endotoxins, can inhibit one of the enzymes, pyruvate dehydrogenase, that is needed for pyruvate to go down to acetyl-CoA and into the Krebs cycle for aerobic metabolism, that is. And if you don't have good aerobic metabolism, anaerobic metabolism can occur via lactate dehydrogenase converting pyruvate to lactate, and another reason why your lactate level can go up during sepsis. And the last cause of this lactate going up is that sepsis can result in microvascular stasis, poor perfusion of tissues, microcirculatory shunting, regional hypoxia, and what happens is then the tissues release lactate. So catecholamine-related increases in the rate of glycolysis, or demands are met by anaerobic glycolysis, poisoning of aerobic metabolism, and vascular changes such as stasis and shunting and regional hypoxia. Those three things go together, and lactate levels frequently go up in sepsis. Some other metabolic and biochemical changes with sepsis: the neuroendocrine response specifically. Sepsis results in inflammation and release of cytokines. The neuroendocrine response includes increased sympathetic activity, release of the stress hormones like ACTH and growth hormone, catecholamines going up, cortisol going up, along with cytokines result in insulin resistance. And if you have insulin resistance, what do you expect to happen? Hyperglycemia. And during sepsis, it's not uncommon to have stress-induced hyperglycemia. Lipolysis is occurring, protein breakdown can occur, and the result of sepsis is a lot of negative things: high blood sugars and lactate, etc. Let's go on to some self-assessment questions just to test yourself over these topics that we just talked about. How might changing a total parenteral nutrition order with the same caloric intake but to a more reduced carbohydrate and increased lipid potentially affect weaning of a COPD patient from mechanical ventilation? So changing the amount of calories in your TPN, raising the amount that comes from lipid and reducing the amount that comes from carbohydrate. Remember that the RQ of carbohydrates, the respiratory quotient, is 1. You're producing a fair amount of CO2 for the amount of oxygen that is utilized, as opposed to lipids, which have an RQ of a 0.7 or lower. So we'd produce less CO2 for the same amount of energy if lipids were supplemented more for our energy source. And that is what can be done up in the ICU sometimes, is adding lipid and reducing the carbohydrate in a patient who you're trying to wean, because all of that extra CO2 has to be exhaled, and if someone is weaning from ventilation, they have to work harder to get rid of that CO2. The next self-assessment question is, why might liver dysfunction result in worsening of a lactic acidosis? Well, that's fairly easy, because I think you know that under conditions of a functioning liver and oxygen present, the liver can take lactate and produce blood sugar from it. Self-assessment question number 3. A patient is pulled from a burning building, and when you draw blood gas from them, there's a metabolic acidosis present, but their mixed venous oxygen is pretty high. What's the pathophysiology of this? Well, one good explanation would be potential cyanide toxicity. Cyanide poisons cytochrome AA3, the cytochrome oxidase pathway, where oxygen is used by tissues to produce energy. If the tissues cannot use oxygen, under aerobic metabolism, anaerobic metabolism can occur, and acidosis the result. Now, if oxygen is not being used by the tissues, it comes from the heart. It goes right past those tissues that doesn't use the oxygen and right back around into the main pulmonary artery where we measure mixed venous oxygen, and it's very high in the 90% range. So cyanide toxicity would be an explanation for this. Self-assessment question number 4. Explain why a prolonged fast may result in the production of ketones. Again, if you don't have a good source of glucose, our body needs it. How can we make energy? Well, we can break down fats, and lipolysis can occur, and the kidney can form ketones, and our body, some parts of our body can use ketones, including our heart, our brain a little bit, and ketones result from prolonged fasts, type 2 diabetics, and low carbohydrate diets or ketotic diets. Those are some of the causes of ketosis. The last one is explain why surgical stress is often associated with hyperglycemia. Well, stress in general results in elevations in epinephrine and norepinephrine, catecholamines, which increase the formation of sugar and make insulin resistance. So that stimulates increase in blood sugar. We have ACTH going up and cortisol levels going up, another reason for blood sugar to go up. And those are just several that are associated with the causes of hyperglycemia that's frequently seen during surgical stress. Let's end there with a quote that is as follows: live as if you were to die tomorrow, learn as if you were to live forever. Mahatma Gandhi apparently said that. So even though this topic may not be the most exciting one, that is biochemistry and metabolism, I hope that you've picked up a couple interesting points that you can take and maybe take into clinical practice. I hope you have a wonderful day.